Hi, my name is Nick Blesnila, and I'm going to give you a brief overview over chapter 376, Traumatic Brain Injury, Animal Models. The goal of this chapter is to provide a brief review of the methods used and the issues surrounding animal modeling of traumatic brain injury, or TBI. Traumatic brain injury is particularly difficult to treat. In fact, we don't have a causative pharmacological treatment for TBI in our hands right now. The reasons are the complexity of the central nervous system and its pleiotropic response to injury, as well as heterogeneous pathological processes in TBI, like different injury severities, different injury locations, and various mechanisms of pathological processes causing tissue disruption and hemorrhage. Currently, mainly preclinical studies have been used to identify possible pathophysiological mechanisms following TBI. Mechanisms are important to explore since they are the prerequisite to develop working mechanistically based therapeutic options for TBI. Here you see the two phases of the pathophysiology of traumatic brain injury. First is a primary injury. It's initiated by tissue deformation during the actual impact. It causes membrane tearing, cell death, rupture of blood vessels, and subsequent hemorrhage. And it is the step which initiates secondary injury. Secondary injury is caused by a sequence of events, by events following each other in a cascade-like manner. Secondary injury, as compared to the primary injury, which takes place within milliseconds, can occur within minutes, hours, days, weeks, maybe even months and years after traumatic brain injury. So the current literature suggests that up to 20 years after a single TBI, pathophysiological processes triggered by TBI are still ongoing in the brain of affected patients. So the main steps in the secondary injury cascades are excitotoxicity, which is the release of or excessive release of glutamate from neurons, leading to calcium influx, cell swelling, and cell death. It's mitochondrial damage, leading to oxidative stress from increasing free radical production within mitochondria. These are also inflammatory responses like cytokine and chemokine release and trafficking of peripheral immune cells into the brain and glial activation. And last but not least, disruption of the blood-brain barrier, causing brain edema, extravasation of red blood cells, and other blood and immune cells into CNS tissues. Finally, the secondary injury cascade results in neuronal cell death and diffuse injury of axons and death of oligodendrocytes and subsequent demyelinization. Right now we have several models in our hands to study the pathophysiology of traumatic brain injury and to test drugs in the preclinical environment. Here on this slide you see two of the most frequently used models. One is fluid percussion injury, or FPI, and the other one is controlled cortical impact, or CCI. In essence, fluid percussion injury produces diffuse axonal injury with a small contusion sometimes, depending how it's performed, while controlled cortical impact is a model for contusions and hemorrhage, which has a small component of diffuse injury, but it's much less than FPI. So FPI is produced by a fluid pressure wave, which is imparted to the brain through a sealed craniotomy, as you can see here. So a pendulum hits here this fluid-filled reservoir and is injected through a small craniotomy into the skull of the experimental animal. And this fluid pressure wave then results in injury, either laterally or here from the top. And by changing the location of the impact, different injury patterns can be produced. So FPI produces diffuse and distributed neuronal and axonal injury with local cortical damage, long-term expansion of lesion volume, and neuropathological consequences similar to closed head injury in man. It can be adapted to study multiple mild brain injuries, and it can be adapted to study repeated mild TBI. And this results in cumulative cognitive deficits, sensory motor impairments, and other changes, which again mimic changes found after human TBI. The controlled cortical impact model requires a craniotomy, a much larger craniotomy than the FPI injury. It uses a mechanical impactor, as you can see here. It's a pressurized air-driven piston, which delivers a trauma with various velocities onto the cortical surface. So the CCI produces a frank contusion and a local hemorrhage. And as I mentioned before, some diffuse injury, but much less than FPI. The model is simple to perform and easy to learn, and also quickly to learn, therefore, and can control temporal mechanical features of the impact. So the CCI model is suitable for longitudinal long-term studies because it has a low mortality rate, and it produces sustained motor deficits, delayed neurodegeneration, loss of tissue, and brain atrophy up to 1 year after injury, and can be used for histopathological analysis and also imaging analysis by magnetic resonance imaging, for example. The two other also quite frequently used animal models for closed head injury in humans is the weight drop injury model. And it comes in two flavors. The Feeney and colleagues model, which is the initial injury model described more than 3 decades ago, and the weight drop model modified by Tony Marmoreau and colleagues. The weight drop injury model, as the name suggests, just drops a weight from a certain height onto the exposed dura of an experimental animal. So therefore, a weight drop is quite similar to controlled cortical impact without the control of the impact parameters. It has been actually adapted from a model with a craniotomy to closed skull injury, so then this piston is placed on the exposed skull. It can also be used with the intact skin, even when the impact is performed directly on top of the head. The problem with the initial version of the weight drop injury model is that it causes fractures of the skull in a very uncontrolled manner. So it's hard to control. Tony Marmoreau and colleagues tried to overcome this problem by just gluing a metal disc on top of the skull and applying the force of the falling weight onto this metal plate. the consequence was that the applied force distributes much more equally on the surface of the brain as compared to the traditional weight drop model, thereby causing much less skull fractures while still causing brain injury. So therefore, the Marmoro, or so-called Marmoro model, produces large amounts of diffuse axonal injury, DAI, which is a very common feature of human injuries. And it can be used to mimic repeated diffuse mild TBI, like to mimic, for example, injuries caused by heading a ball in soccer or by repetitive head traumas in American football. And it produces long-term cognitive deficits, which is also very comparable to that what we observe in humans. If not acceleration, deceleration, or impact injuries are investigated, but for example, penetrating brain injury, then other models have to be used, like the penetrating ballistic-like brain injury. It produces injury by a penetrating rod or an elastic balloon, as shown here. Therefore, it mimics injury caused by bullets or other projectiles. It produces a high-velocity penetration of the brain with a lot of hemorrhage and tissue destruction. And it's mainly used to study injuries produced by bullets in the military arena, for example. Another injury model, which is also often used to study military-related brain injuries, is a so-called blast injury. So this blast injury TBI model doesn't produce an impact directly on the skull, but it basically uses a pressure wave or a blast wave to produce several injuries on the whole body of the animal, but also on the brain. So it produces a percussive wave initiated by a blast at a distance from the animal. And it's designed to study the kind of injury seen in military personnel exposed to explosive devices. It is very useful to study emotional reactivity and post-traumatic stress disorder, PTSD, which is very common in human blast injuries. More recently, the interest of TBI research shifted a little bit away from military injuries, penetrating injuries, and acceleration, deacceleration, and impact injuries to mild traumatic brain injury, which actually affects about 80% of all patients suffering a TBI. So 80% have a mild injury, and this was under-investigated over the last decades. So here in H, you see the closed head impact model of engineered rotational acceleration with a very nice acronym CHIMERA. So it's a newly developed device. It's non-surgical and enables kinematic analysis of unconstrained head movements. And it doesn't require a lot of anesthetic. So the animal is placed head down on this platform, and then basically the animal is moved down so that a rotational injury is induced. The other device recently developed, which is also non-invasive, is a closed head projectile concussive impact, PCI. It has been developed to mimic the injury from war zone and multiple concussions from sport-related activities. It's also a device which has to be purchased like the CHIMERA device. What we didn't discuss so far is here under G, the combined injury and polytrauma. So all the models just described can be combined with polytrauma. 50% of traumatic brain injuries affect only the brain, but the other 50% often occur in combination with polytrauma, so for trauma to other parts of the body. So what can be done is to incorporate one of the TBI models just discussed, along with the physiological manipulations such as hypoxemia, hypotension, concurrent fractures, spinal cord injuries, peripheral injury, or repetitive injuries to mimic the effects of repeated concussion. These models, however, have multiple limitations. First of all, ethical issues, because the animal has to be injured in multiple ways. And usually these models fail to produce consistent injury outcomes in rodents. So it's problematic to perform multiple injuries in a mouse or in a rat, specifically when one aims to investigate long-term outcome after these polytrauma models. Equally important when considering which animal model to use to study TBI are outcome measures, specifically mild traumatic brain injury, where the impact may be so mild that impairments may be very difficult to investigate. It's very important to have the right outcome measures. First, we have physiological responses like intracranial pressure, respiration, oxygenation, which are changed mostly in cases of severe TBI. The same is true for motor cognitive function or dysfunction, spatial memory impairments, and changes in social behavior. One of the most frequent outcome measures are anatomical and histological measures, like lesion size, for example, by histology, or by imaging like MRI or PET, and by quantifying the amount of cell death by counting cells, of axonal integrity, by staining axons with different antibodies, and of course by looking for inflammation by staining inflammatory cell invasion. As mentioned before, however, sometimes none of these single readouts will give you a robust outcome measure. And the solution here, which was recently started to be used in traumatic brain injury research, is machine learning-based data integration. And it's machine learning using common data elements is possible to perform data-driven multidimensional analytics to identify consistent patterns of association across biological and functional endpoints. And that's what you see here. So each of these single readouts, like incline beam walk, grid walk, and some histological parameters, actually do not give you a consistent readout in a given model. However, when you combine all these findings and you integrate all these findings into a multidimensional pattern, you may well, as you can see here, see very large differences with a very reasonable standard deviation between sham-operated animals and traumatized animals. So this enables rapid development of targeted experimental studies to validate robust pathofunctional endpoints and find translational targets more quickly than before. So the conclusions of this chapter are that different TBI animals models generate their unique pathological outcomes, which can provide the most appropriate animal model for addressing a specific hypothesis and mimicking pathology in human TBI. Animal models provide a continuing and rich source of new therapeutic targets for TBI, and continuing improvement of these models is necessary for the successful translation of preclinical findings to the clinic. To improve the rigor of preclinical and clinical trials, common data elements, CDEs, have been developed, such as the Federal Interagency TBI Research, FITBIR, and the Open Data Commons for TBI infrastructure. These efforts are aimed at accelerating harmonization among preclinical TBI data, thereby increasing scientific reproducibility. the common and continued back translation of data and concepts from the clinic into animal models of TBI is likely to enrich the field and improve the changes for translating laboratory findings to man. Thank you very much for your attention.